Hi, everyone. Thanks so much for joining. We'll just wait a minute to let some people into the Zoom room. Seems like people are still coming in. All right, let's go ahead and get started. So hi, everyone. Thanks so much for joining our webinar today, which will be on essential techniques for culturing brain organoids. My name is Afrida Rahman Enyart. I'm your host for today. I am a product manager at Proteintech. And our speaker today will be Michela Barbato, who is a senior research assistant at University of Edinburgh. Before I pass it off to Michela, I just want to share a few company slides to introduce Proteintech to you. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a very expansive portfolio of antibodies that encompass over 13,000 targets and cover many different research areas. We also offer ELISA kits, IHC kits, and antibody labeling kits. Within our Proteintech brand, we also have two other lines. So we have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells. And because they're expressed by human cells, you can be confident that they have native folding, post-translational modifications, and glycosylation. And the third brand that we have is our Chromatech branded nanobody-based reagents that are based off of alpaca nanobodies. And as they suggest, nanobodies are very small antibodies, so they're really ideal for clean immunoprecipitations, super resolution microscopy, and protein purification. I also just want to take a moment to celebrate the success of us and our customers. So Proteintech products have been cited in over 200,000 publications worldwide. So we're really proud of that. And we're really proud that we were the winners of the Researcher's Choice Award last year from SciAb. So with that, I'd like to introduce our speaker for today, Michela Barbato. So Michela is a senior research assistant at the University of Edinburgh in charge of the Edinburgh Progenitor Cell Bank under the care of Professor Thilo Kuhnhoff. Previously, Michela had been in charge of the brain organoid facility for more than 4 years under Professor John Mason and worked as a stem cell scientist at the UK Stem Cell Bank before moving to Edinburgh, where currently she is working with differentiated human iPSCs and human ESCs to study dopaminergic neurons in Parkinson's disease. So with that, I'll go ahead and I'll hand it off to Michela. Thank you so much for being here today and for sharing your work with us.
Hi, everyone. Thank you, Afrida, for inviting me. I'm just going to share my screen for my presentation. Just 1 second. Here it is. It was on the wrong slide. That's why I needed to go back to the previous one. So, hi, everyone. As Afrida give me a nice introduction, I'm here to talk about how to culture human brain organoids. And I've been working with them for more than 4 years. And we troubleshoot the way how to culture them. And so I'm here to just give you some tips and techniques for it. Just an introduction about organoids, especially brain organoids. Since 10 years ago already, Lancaster came out with this protocol where it was possible to lift stem cells and they were self-aggregating, so to form this structure where further on was called brain organoid. After that, many other labs, and I'm here to cite the Pascal lab, which also works on organoids, developed other protocols for differentiate the RPS cells in organoids, and in the picture you just see one of the organoids from my lab. So how do we get there? Of course, we use pluripotent stem cells, and the way to approach it, how do we differentiate them, of course, is to choose a robust protocol from the literature. In our lab, we decided to look into this different protocol, and where they were generating cerebral organoid mainly. So pluripotent stem cells, of course, they can be embryonic stem cells, so they derive from an embryo, they've been cultured and afterwards they've been isolated, or you can get induced pluripotent stem cells from reprogramming some specific type of cells. Which this can be somatic cells, so fibroblast or blood cells, where you just put the Yamaka factor and then you derive the iPS. And the iPS, of course, they are very useful because you can differentiate in different subtypes of cells like neurons or other type cardiomyocyte if you're different. It depends what's your study on. One thing about induced pluripotent stem cells, of course, it reduced the fact of some restriction, because some countries they might have issues with embryonic stem cells. So when this was discovered, kind of made things very helpful for differentiating different type of cells. So for pluripotent differentiation, as you see, we have two different types mainly, the 2 differentiation and the 3 differentiation. So I'm going to just briefly introduce you why also 2D differentiation is important, and then I will move on the 3D part, so actually the brain organoids. So as Frida mentioned, I work as well with 2D differentiated stem cells, where in our case we try to make dopaminergic neurons. The idea is that with the stem cells, to differentiate them in progenitor cells. And the good thing about these cells, they can be banked, and later on in different stages, they can be thawed out and differentiated, matured in mature neurons, exactly, where you can do drug screening and human in vitro studies. So that's at the moment what I'm also working on, where you basically, you can differentiate your cells. We differentiate them, we do the complicated step, we cryopreserve them, and we just distribute the bank of progenitor cells. That's because, of course, working with stem cells, it can be very challenging, and many labs, maybe they don't have enough expertise, or maybe they don't realize how expensive also is to conduct studies using embryonic stem cells and stem cells. So this is kind of like a help for research to move forward, especially if you want to study neurodegenerative diseases. With saying that, I want to also mention that to differentiate either neurons or brain organoids, what is important is the quality of your stem cells, because if your starting point is not good enough, then it's very hard for you to get to the next steps. So you want to make sure that all the line that you're using is able to differentiate in all the germ layers, because sometimes might prefer to be endoderm or ectoderm. So there might not be the cell line that is useful for you. And also you need to have the quality of your iPS under control. So you don't want spontaneous differentiation in your culture while prior differentiated into organoids or to these cells. So I'm saying this because we've been very lucky in our lab at the beginning when we set up the facility, that we received Indonesian line, and unfortunately we didn't know at the time, but we worked on it for a few months, tried to differentiate them in brain organoids, but as you can see from the picture, we didn't get any success from it, and we realized that actually this cell line was used to make blood cells. So then we were wondering, okay, maybe not all cell lines are good to differentiate in brains. So something that I learned when I was working with the UCAN Stem Cell Bank, it was always to kind of give this embryo body differentiation assay to our clients, because knowing what type of line you're handling is very important for you to go towards a certain type of cells. So in this case, if you are prompt to do brain organoids, maybe you want your neuroectodermal genes are highly expressed. Also, for keeping your stem cells under control, you might want to do routinely some stem cell marker assay, so that you're sure that your NANOG, OCT4, and SOX2 positive cells is still high before differentiating your cells. So saying that, of course, we can distinguish two types of protocol for growing brain organoids. One is undirected differentiation, as I mentioned from Lancaster, and the other one is direct differentiation, where you actually choose to pattern your cells towards a specific fate. And this came out from the Pasca lab in US. So the indirect differentiation is basically lifting the stem cells, let them self-aggregate together according to the patterning that you're giving them. But in this case, it's like just self-patterning, so they go spontaneously, almost going towards like a brain type organoids. The technique is basically, as I say, to lift, form embryo bodies, give some neuroinduction media, then at some point you embed in matrigel, and you will let differentiate for a while and put them in a bioreactor, so that the organoids, while they grow, they get all the diffusion of the nutrient of the diffusion inside. So as I said, this is from their paper, from the Lancaster paper, where you can see all the steps where you have the stem cells, then bit by bit they get a bit bigger, the embryo bodies, the matrigel step, the bioreactor, and how you can see a rosette from an organoid. And also these are some stainings done from it, so that it showed you actually this organoid that there is in the dish. It has different subtypes of brain cells. So there are forebrains, choroid plexus, and hippocampus in this case. In the direct differentiation instead, for example, in this case, it's a protocol for cortical forebrain. So you would put your cells towards this pattern, and you would expect it to have exactly just cortical cells. Pascal was also what did in his lab. It was also very good in finding another way to pattern their cells. So they were also kind of ventral forebrain. So I only did the cortical, but also did the ventral one. And what it did at some point, it just aggregate together so that they could make assembloid. And this is very helpful, because if you're interested to know what type of communication can happen in two different parts of the brain, is kind of a good way to study it. That you have a part that is forebrain and the part is ventral, and you can see the migration of the different interneurons, and that's what happens during the stains, where of course is showing that the forebrain doesn't have the ventral markers, while the ventral markers, they don't have the forebrain markers. And then once they combine together, you can maybe start to see how they interact with each other. We did use both protocol in our lab. So we did use the indirect differentiation and the direct differentiation. With indirect, we were a bit more lucky, but with direct differentiation, we had a lot of issues, because our organoids, they kept fusing together and they were not really happy in those situations. But we did believe that patterning our organoid towards the forebrain, because that's what we were interested mainly. So that's what we decided to compare them and see what we can take. Let's say we took the best out of each protocol, and we basically troubleshooted and see what was working for us. So something that we observed, of course, it was that one protocol had a matrigel embedding and the other one didn't. You would think like, oh, it's a step less, so why should I worry about the matrigels? But we thought that maybe it was something important to look at it. Also, the shaking and not shaking, that was also another step that we took under consideration. The timeline, also when you want to fix your organoids, is also very important, because you want to make sure that your marker of interest marker, they are there when you actually fix your organoids, because we are talking about growing organoids for more than a month minimum. So you don't want to waste your time and then find out that the markers show up at a later stage, and then you need to restart from the beginning. Exactly, that's the difference between the 2D and the 3D, that unfortunately the 3D, there is no still a way to freeze down the organoid and bring it back. Also, we were wondering if there is a difference with the seeding of the cells. So do you need to seed them in a specific concentration? And also, why do organoids fuse together, and how can we avoid that? So in our lab, as I said, we decided to kind of troubleshoot it, and we say, okay, let's take the steps, some steps from one protocol to another. And we decided to lift the cells, and we seeded a specific concentration to create a base. And then we were moving them from a 96 well plate in a 24 well plate, where each well had a single organoid. And on a certain date, we were embedding in Matrigel and putting on the shaker. But the first part was like on a slow shaker. And then it became, when they were on the mature phase, then we would have put it on a faster shaker, because at that point, the organoid is quite big. And you want this, again, the nutrient to reach as much as possible the core of the organoids. So it's just a chart with more details. So what we're doing, as I said, we lift them, we put the neuroinduction media, we embed them in Matrigel. Even there are different techniques to do it. We troubleshoot our way. It seemed to work. It's kind of tricky. So that's probably why maybe when you see the part that is not Matrigel coated, the organoids is like more appetite, but actually it does make a difference for us, at least for our organoid. And then at the later stages, we would do this maturation with adding the BDNF and NT3, and then at some point we would remove them. We would just keep them on culture media so that they could just grow until our fixing time was due. So bringing up together, we saw that the seeding of the cells, according to specification, helped us to make our organoids grow healthier. The quality of the plates also was very important for us to find out, because if you use a plate that it has just a normal culture plate, your cells will tend to attach. So what we did was to use those low adherent plate. They're a bit more expensive, but they really work if you want to make your embryo bodies. So we did use those plates. And also what helped for the fusion, as I explained, it was to separate the organoids in a single well. They're adding the right growth and differentiation factor is also important, because if you want a pattern towards a specific part of the brain, then you want to make sure you're using the right factor for it, that you choose the right pathway to activate it, basically. Matrigel helps the growth, but also we noticed that it creates a kind of like a scaffold, so they tend to disintegrate less, the organoids, when we were growing them. And also the shaking platform really helped for the diffusing of the nutrient. Another thing that you need to think about when you work with your organoids is the timeline, as I said. Of course, we're trying to compare the timeline as it would be a normal embryonic stages, but it is not, because we go from embryonic stem cells, like from pluripotent stem cells, we differentiate them. And of course, the markers, they show up in different moments compared to like a normal phase, like in vivo phase. So there are many charts out there that you can compare a bit, like, for example, at day 60, what is the gestational stage of your brain? That kind of helps as well to realize what type of markers you can be interested in finding out. So I really suggest you to keep an eye on those charts as well, in case you want to, you are interested in a specific gene and you want to find out when it shows up. Now I'm gonna go showing you a series of organoids that's what we said from our lab. These are for control that we derived from a project that we were working on, where we wanted to knock out the PAX gene. So this is day 1 organoid. So it means the day after of seeding. So when they start to aggregate and they form the embryo bodies. As you can see from the first day, you might not see much difference from them because they're just aggregating in the size. But at day 7, when we kept in culture, we noticed that between the 4 of them, the actual 4, the control 4, was a little bit smaller than the other one. As I said, these are coming out, they are control clones coming out from a clone selection. So you would expect some differences between clones. At day 30, after embedding in matrigel, we could see that the organoid was actually interacting with the matrigel, and the structure was actually growing bigger and faster. But as I explained to you, the control 4, it looked really bad. Like even in matrigel, it just didn't succeed. So some lines, they might need more, more trials in making them, or, it doesn't mean that this one was, because the parental line is the same. So we use this parental line for all our brain organoids. But basically, this one maybe was just a bit of a failure for this trial. So we repeated it and we went forward with that anyway. So a day... And later on, these organoids, you can see under the microscope that they are forming some rosettes. So, for example, here you can see actually some rosettes from like rings showing up in these pictures. So you know that actually the patterning is also working, and your organoids are going towards the fate that you were wanting. And at day 44, you can see even more how big they are, and also the expression of this rosette is even higher, so it's even clearer. We pushed a bit further our organoids. We grew them up to day 135. We didn't try to cut them much. It was more like more to see if they could grow at that stage. They can. That was our first trial, basically. And that's the size of my thumb, to make you understand how big is this organoids at day 135. So we did the afterwards section. These organoids I was showing you, as I explained to you, you can see here from day 30 until day 60, the difference of the expression of these two markers, CTP2 and SATB2. And also here that we have this organoid like showing up. And this is basically the organoids I was telling you about, that we grow them from this experiment that we were doing with the knocking out of the PAX-seed genes. And we finally kind of have in bioRxiv a paper that is under review for knowing more about this story. So in case you are curious about what's happening if you delete PAX6 in organoids, you can check it out. Also, something that we thought to do, it was to, okay, we have these organoids, and we were interested to understand the connection between thalamus and forebrain. At the time, we didn't have much experience in making thalamus organoid, but we did have the possibility to get thalamus from mouse. And we used this green mouse that it was stained for GFP. So with our organoids at day 18, we sliced it, and then after we co-culture with a slice of mouse brain, and we noticed that from the part of the mouse, which I said is green, there were some ejection coming out that they were going towards the organoids. So that's also a way that you can use your organoids between in vitro and in vivo study. You can do some co-culture experiment with it. So saying this, of course, you have your organoids that you want to use all the information from it as possible, because as I explained, it takes a long time to grow it. You want to make the most out of it. So before you start your culture, you really need to understand what's your final question from it. You cannot just grow them and then at some point stop and then realize that actually you need to go further down the line. So organoids, they're very good if you want to do immunostaining. If you have the chance to do 3D live imagings, they are very good as well. Once you do immunostaining, of course, one technique to use is to understand like what the quantity of markers, so how many cells are expressed in the marker that you're interested at. I know it's very painful, but it can be done through cell counting. Also, qPCR analysis is quite useful to understand what's going on in your organoid, and single cell RNA-seq is also very important, because in that way you can just dissolve your organoid and you can just see what type of situation, what cell type are present in the organoids. Because even though you do forebrain, it doesn't make sure 100% that you're just having 100% forebrain cells. Another type of organoids that we grew in our lab was the ventral organoids. We did receive these lines that they had the control and the deletion line that it was for the gene 1611. So basically, we did culture them till day 50 and we investigated this gene that is called LHX6, which is a ventral gene, and we realized that the comparison between the control and the deletion line at later stages, there was a decrease of this expression LHX6. Also, we did another test, like it was to check the GAD67, which is for interneurons, and we found that it was present in this organoid. So at least we were sure that the organoids that we were growing, they were ventral organoids. And we did qPCR data analysis, and even there we saw that the organoids, the expression LHX6 was decreasing over time in the deletion line. We did also some single cell analysis, but a nice paper just came out this year, beginning of this year, regarding this topic, where there is an explanation, and it seems like the 1611.2 deletions cause like a early maturation in organoids. So if you want to know more about that, please reach out. And also, I'm taking just the opportunity to present also this project has been done with Rana Fethith, where she's gonna present also as well in another webinar further on the year. And so in summary, I just wanna say what I've been talking for now. Of course, the quality of your hPS is very important so that you can differentiate a healthy brain organoid. Culturing your brain organoid can be very challenging. If you are all alone, please try to find some help as well, because it's very demanding. So not only much the stem cell part, because once you differentiate them, you can stop your culture with the stem cell. But of course, if your question is very it goes down the line, like you need very mature organoids, then it means you need to stay for months culturing them. We did have some I helped also another lab that they're trying to take the organoid like in a postnatal phase. I don't know if they managed, because it was quite impossible. I tried just to help them until the early stages, so until the 30, I helped them train them, but then I don't know if they went further with that, but it's challenging. Also, data analysis, of course, it can give you a good answer. Again, if you have the right question, you might find out what's going on in the brain organoids. And there are no good tools for studying normal and diseased brain development. So definitely, if this is something that you would like to work on or you're working on, it's something that you could look into it and see if it works for you. With that, I just want to thank the labs that were involved in this project, and thank you for listening to me. And if you have any question, I'm happy to answer them.
All right. Well, thank you so much, Michela, for your wonderful talk and for sharing all your insights on brain organoids. Before we begin the Q&A, I just want to share a few product slides that might be interesting for our audience today. So at Proteintech, we offer humankind growth factors that can help with the maintenance and differentiation of your brain organoids, whether you're looking at whole brain organoids or a specific region of the brain. Additionally, we also offer antibodies against different cellular markers, such as progenitor or neuronal markers, that can help you characterize your organoids. Um, So in addition to having key proteins that are necessary for organoid differentiation, we also have noticed that our recombinant proteins perform better than our leading competitors. So here on the right-hand side, let me just get my pointer going. Here on the right-hand side, you can see an example of our recombinant Wnt3A compared to a leading competitor. So on the top graph, we measured luciferase production in response to increasing concentrations of recombinant Wnt3A for 6 hours. And we observed that Proteintech Wnt3A has a threefold lower EC50 and greater induction of luciferase production than our leading competitor. And this suggests then greater activity. Additionally, when comparing with our top 5 competitors, we've observed that our Wnt3A protein has higher purity. So what are the main benefits of using our humankind proteins for your organoid cultures? So the main advantage is that our humankind proteins are authentic human proteins because they are raised in human HEC293 cells. So they have, native glycosylation and protein folding. Additionally, we have seen that they have superior bioactivity, as I showed you on the previous slide, using our Wnt3A as an example. So this means, enhanced results and healthier cultures. You can also be confident that our proteins will be animal component free, xeno-free, endotoxin free, tag free, and carrier free. I also just want to talk a little bit about, why the expression system matters so much and, why at Proteintech we put so much emphasis on the fact that our proteins are derived from human cells. So here in this figure, we're looking at a comparison between our humankind TNF alpha and TNF alpha that is derived from E. coli. So E. coli-derived TNF-alpha tends to be expressed in monomers with no post-translational modifications, high endotoxin levels, and improper protein folding. Whereas in our humankind system, we see that our TNF-alpha is expressed in trimers, which is the most active form of TNF-alpha. And we have, proper post-translational modifications, proper protein folding, and this all mimics what you would observe in human TNF-alpha. So you can really be confident that, the protein that you are exposing your cells to is equivalent to what you would observe in a human system. And I just really quickly want to show this customer data that we received. So we had a customer that used our humankind proteins in their neural stem cell cultures in order to look at differentiation of dopaminergic neurons. And they were able to observe, as you can see from this beautiful staining and image, they were able to observe markers that confirmed the differentiation of their stem cells to dopaminergic neurons. I also want to highlight that if you are looking to differentiate your stem cells into neurons, Proteintech does offer BDNF, a key protein that's needed for this differentiation. So we've observed that our BDNF has a greater potency compared to competitors, and that it results in better neuronal differentiation, as you can see in these comparison images. Uh, using a neuroblastoma cell line. So you can see that the cultures that were treated with our humankind BDNF have, more extensive neurites compared to those that were treated with the competitor BDNF. And then lastly, I just want to mention that in addition to our humankind proteins, we also have a large portfolio of antibodies against neuronal and progenitor markers so that you can characterize your organoids. So here is just a few IF examples highlighting our PAX6, SOX2, and TUDG1 antibodies. And we do have antibodies against, over 13,000 targets, many of them covering targets for the field of neuroscience. And as an attendee of today's webinar, you will receive a follow-up email, which will include a recording of this webinar. You will also receive a certificate of attendance, as well as a link to request a free sample of one of these humankind proteins, so either BDNF, noggin, activin A, or GDNF for your organoid cultures. We do ask to please allow 1 week for this follow-up email to be sent. So with that, I think we can go ahead and begin the Q&A session. If you do have any questions after the Q&A session is over, feel free to email us at events@ptglab.com, or you can directly reach out to Michela at this email address too. So with that, we have some great questions that have come in. So we have one question. So actually, there was a couple people that were interested in this. With organoids that where you're growing them for a very long period of time, like 135 days. Do you have to deal with necrotic cores? And if so, how do you kind of deal with that and manage that?
Well, that's the thing. As I said, for our very old organoid, we didn't really try to cut and stain, because actually for us it was more like to see how further we could push, because also our question were early stages. So we maximum reached day 90. So but day 90 is already old enough. The necrotic part is always like something that puzzle whoever grows organoids, because it's like, is it really necrotic in the middle, or you actually have some cell type that maybe you didn't know about it? Because when you do single cell analysis, as I explained, you might find out to have different part of the brain showing up. But the idea is that you will find always a bit of necrotic part, but the difference, the helping of the shaker is just that you reduce it quite a bit. Of course, we didn't do any statistical analysis and try to understand how much the diffusion on a shaker would help, but that's why you go from a slow motion to a bit faster motion, because your organoids will consume your nutrients like in less than 24 hours when they are at that later stage. And something I didn't mention, I kind of forgot, that once we are from a 24-well plate, around day 45, we move them in a 10-centimeter plate. So you will put at least two, 3 organoids together. At that stage, it's fine because they won't fuse anymore, but still you would have like, it's also to save space on a shaker. That's why you put a couple more inside the same plate. But they become yellow quite fast. So at least every other day you need to feed them.
Okay, great. Thanks so much, Michela. The next question that we have is, did you perform molecular analysis, gene expression to examine the effects of the matrigel?
No, we didn't. As I said, we were mainly trying to work out the focus of what's happening, like, first of all, if we could grow organoids, the second thing, if we could get the cell type that we wanted. But I know that few labs, they actually removed matrigel afterwards, because they have the fear that the matrigel could interfere with the, because it's like a mix of growth factor in it, so you don't know how much is affecting the growth of the organoids. And with this, actually, as a picture I showed you, you can see that the organoids actually interact with the matrigel. So it is puzzling what's actually happening when you put the matrigel in the organoid. Unfortunately, we didn't have time to answer that question, but maybe other labs, they're doing it right now. But as I said, some of it, they remove it, but we thought it would have been quite tricky to do it. So we let them grow until the matrigel completely disappeared from the organoid.
Great. So our next question, again, kind of discusses about the fact that, these organoids are being cultured for such a long time. Do you have any issues with contamination? And if so, how do you manage that?
So we cultured them in antibiotic and antifungal. We did culture them in that. but still it doesn't guarantee you that you don't get contamination. So of course, a very good aseptic technique is what is really important. Again, you're handling plates that they can open very easily, and also when you are removing the media, you need to be very careful with that. So for sure, good aseptic technique is what it's a must, and what helps as well is also putting the antibiotic in it. Let's say we didn't have a breakout at all, but we did have every now and then one plate that was just getting that random contamination. But that's why you try to not do the full 96 word plate, but at least to calculate that maybe you can get contamination at some point. So maybe you do 20 organoids, 30 organoids. So you're on the safe side.
Awesome. So our next question kind of discusses more about kind of quantifying and looking at the size of the organoids. So did you measure the organoids to compare the different controls? So how do you do that when the growth of each organoid is not uniform? So for example, like they aren't spherical.
Well, the idea of seeding at a certain concentration, that it helps as well, because you know the starting time, like they're all seeding. For example, we all see them at 9,000 cells per well. So at least you know that at the beginning, the growth of all embryo bodies, they should be all the same. And also because you kind of, I mean, it varies, of course, because there are different organoids. They might have a bit smaller, but the majority of them, they should be on the same size. They should be the same size, unless you're comparing control with, as I explained to you, with the 16P 11.2, you would have different sizes because there is actually a deletion, there is a mutation there, and that gives trouble. But when we wanted to look to size, we did do analysis with software when we were kind of calculated the diameter of the organoid from the plain picture that I showed you under the microscope, not the one with markers or anything. But we did realize that there was a difference. And you can see by eyes as well the difference when it's like a mutated line compared to a control one. But generally in a batch, you will have variety, but it should be more or less the same.
Okay, great. Thanks. We have a couple questions from Amal here that are more technical questions. So the first question is, how important is the shaker RPM for organoid culture? So have you observed any differences in organoids cultured at 60 versus 90 RPM? So we'll start with that, and then I'll go to the second question.
Yeah, so we use 45 and 90. We didn't use 60. But we did see the difference from not having at all. So the Pasca protocol doesn't need a shaker. So when we were trying to grow the organoids, not using the shaker, we saw that they were much smaller than the one they were on the shaker. And that's because when you shake them, the diffusion of the nutrients gets inside the whole organoids faster. Instead, when they are static, the organoid is just maybe getting on a side, on the other side, but they were very small. The speed, it changes because you go with the higher growth. So the bigger it gets, the faster you should go. Again, because they use nutrients faster. So that's why we go from 45 to 90. It's not much the difference. It's just a match of for them getting the diffusion, a better diffusion.
And then the second question is, have you observed a great variability in the number of neurons versus glia across organoids cultured from the same batch of hPSCs?
We didn't look into glia cells because, as I said, we were interested very early stages. So I think glia cells, they will appear much later. The maximum that we went for to actually stain and investigate our organoids was day 60, day 90, but day 90, very little from it. We did a lot of work on day 30 and day 60, both for brain and ventral organoid, actually. So we didn't look into other cell type.
Okay, Great. So we have another kind of technical question about, how do you break down the organoids in order to do cell counting?
There are different, so the actual organoids, I guess, if you want to know how many cells there are in an organoid, I guess that's the question.
Yeah, it seems like that's the question.
I think you do that if you want to do single cells RNA-seq. So you need your organoid to go in single cells. And there are products out there that there are like, they help you to dissociate the organoid, they reduce them in single cells. There are protocols to do that. And the postdoc in my lab, because he did single cells RNA-seq, he just used that product and he basically dissolve the organoids in this in single cells, but there are ways to do it.
Right.
And do cell count, because once you get them in single cells, of course you can do cell count with a cell counter.
Yes. So there's a question here that maybe you have some insight on. So, how do you explain the fact that, some human PSC lines are able to differentiate into cerebral organoids while others are not? And is the heterogeneity between the different control lines a problem?
So, as I say, that I think because we're working with embryonic stem cells or pluripotent stem cells, they kind of go default towards, one type of subtype or another. That's why with the UK Stem Cell Bank, we were doing a lot of this embryonic embryo body differentiation assay, because we were providing stem cells for the public for other labs, and we wanted to make sure that if they wanted to use it for a certain study, they would be able to differentiate it that way. The reason I'm not sure if I can only think that, in nature we do have stem cells that they go towards one type and towards to another one, and once you isolate them or you reprogram them, then it means that some part of that knowledge gets into them and they will go more towards a different part or another. As I said, sometimes in a matter of being fast, because you are underfunding, so you need to produce data. So when you have a line that really is not working, you don't waste too much time in it in trying to understand why you're not working with me. You just go, okay, the other line works better, so I'm just gonna stick with it. So, but it does make, I think, the difference. Like, it's good to know, but if you have one line that works, I would not question much why it works and the other one not. I would just go for it. But I think it's like just a natural inside of the cells that they go towards a fate or another.
They just have their own personalities, and some are more susceptible than
I mean,
others.
if you work with stem cells, you call them baby, you call them all sorts of stuff. You're like, why are you angry at me? Why you decide to die? It's just literally like a life lesson working with them.
It's
So,
exactly
but
like toddlers, like, just
yeah,
talk
they're
to me.
toddlers. Yeah, I mean, with I just say like this little background about this work with the 16p11.2. I was the only one able to grow the stem cell. Like every time I was going to go on holiday, I could not leave because literally I was receiving a message at the plane that the cell just lived to die. Like I was not doing anything any difference. Like all the people that were training the same way that I was. I mean, I trained them, so it's not that they were doing something different, but as soon as I was on the plane, they were like, yeah, she's gone, I'm gonna die, so
They realized their mom had left. They were
yeah,
rebelling.
so that's the thing. Yeah, I don't know, operator is also something that it's a key. For example, in my 2D culture, it took me a while to get the hands on it, and now it's like, it's not that I don't know how to culture stem cells. It literally is you finding your way how to make the technique work with your way of working. So yeah, that's that's the thing.
Great, thank you. So we have a few more questions. So there's a question here from Patricia asking, do you think that the microfluidic devices may be a good way to produce organoids?
Macrofluid devices. I'm not very familiar with that. but I guess they could work. I guess like replacing from shaker, something like that. I guess. I guess it would work. As I said, it depends. As I said, I don't know much about the microfluid. I don't know really how they work. Uh, but I guess they could, if you can manage to keep them separated, like if they have chambers where you can put your single organoid, maybe it could be helpful. If you work with that, I would try to give it a go and see, if you're an expert in microfluids, yes, you can
Yeah.
try.
And maybe there's some literature on it. So
Yeah. I
maybe,
mean, there are different, I mean, the organoids have been out there now for 10 years. And so it means that for sure someone is working on it, as they were trying to figure out a way how to diffuse better, because of course organoids, they don't have endothelial cell, they don't have vessel, they don't get blood, they don't receive oxygen. So they're all trying to co-culture, with endothelial cells, so they can see if something, come out of it, and they can get oxygenated without you going and shake them, So yeah, I think we will probably know better in a few years about that.
so there's another question asking, so do you tend to see nuclei within the organoids like you would in the brain, or is it more general where the cell types are present, but the organization of the nuclei are not quite the same as in vivo?
I'm not much familiar for in vivo studies. I haven't much worked with tissue coming out from actually, animals. But when you cut an organoid, then, for example, the first thing that you do, you would mark with the DAPI. DAPI distributes to organoids like everywhere, even in the necrotic part. That's why also there is a debate where the necrotic, it's really necrotic, because DAPI is there, is reaching the nuclear. But whether it's dead or not, if you do some staining with, I think, Cas9, you won't find much in the middle that it's dead. So literally the nuclei doesn't make much. It's not a direction for you to know what's the difference. I mean, at least in the organoids, of course, if you compare in vivo, there is a totally different structure there. So there might be some difference because it's a whole brain, for example. Instead, there you have just a part of a brain. So I guess there is a difference.
Yeah. So you had a lot of, really beautiful images in your presentation. Do you have any tips in terms of, how to stain your organoids, like sections, magnification, anything like that?
We did fix in PFA. When they're small, there's not much problem. You can just do 4% PFA and you follow any protocol for, fixation. I mean, I'd fix the 2D cells the same way that I was fixing my organoid. Maybe you just leave them a bit longer and shake it, because of course, you always need that the PFA goes all around the organoid. The dissection, we were using a cryostat, and we were also doing a certain degree. So we were doing like I think it was -20, -23. It depended of your machine. Ours was very old, and I hated it because it was so cold. But
Mhm.
but we
But they never
did.
want to get rid of it. They don't care how old the
No,
machine
no,
is if
no.
it's
I
not
was wearing
broke.
like three pairs of glass trying to dissect my organoids, because also when they're small, so when we were fixing them afterwards, we were embedding them, and it was a solution, half sucrose, and something else. Sorry, I don't now I can't remember which, but I have the protocol in case you are interested to know how we were doing in details. But what we were doing in the actually mold, we were putting more organoids together if to fit, so that when we were cryosectioning them, we knew that, those were our 4 samples or 3 samples, and we were putting in a series, so that when we were going, like starting to cut, we would know what was the beginning, what was the end of each one of them. So that's how, and then we were just immunostaining like with any protocol for immunostaining, maybe doing some antigen retrieval for some marker, but it was very basic immunostaining, and then you go to the fluorescence microscope. You will add your section on a slide, so of course you wouldn't do in a culture plate. You will keep doing like on a slide, on a microscope slide. Yeah.
Well, thank you. So we have time for maybe one more question. So there's a question here from Thomas asking, when you started organoid culturing, what did you find were the biggest hurdles to overcome?
The big obstacle for us, first of all, of course, it was like to choose the protocol, because we were like, okay, which one are we gonna go first? Then when we decided which one to choose, the knowledge behind it was very little. I was an expert in stem cells. I was no expert in neuroscience. I don't have any background in neuroscience. So for me, the patterning, it was completely clueless. That's why the postdoc kind of helped along. But I guess the troubleshooting of the technique, that was very hard to get over it. It took us a while. It took us 6 months, more than 6 months to get it on. But once you get your hands on it, then it's like you know what you're doing. You know that your organoids, maybe you can fail a batch, but you can do the next one the next day, and that one is fine. Of course, the variability also is something that it really frustrates you at the beginning, because you're like, what am I doing wrong? Why this batch is not coming out, and I did one yesterday, it was fine. So a bit that is the frustration that can come out from growing them. But the beginning was more to understand what sort of technique to use, like especially, for example, the matrix gel. That was like, because you are facing expensive reagents, and you don't want to waste them. So in the moment that you are embedding them, you need to really know if they actually need it. And I know that some labs, they were putting matrix gel even in the media. So not only you were embedding, but you were also putting matrix gel in the media. So it's like, is it really worth it? And in fact, we didn't put matrix gel in the media anymore. We were just embedding and stick with that. And it seemed to work at the end. But I think the beginning is always hard for getting the everything going on. Yeah.
Yeah, absolutely. And as you mentioned, they've been around for 10 years, but in the world of science, that's like not very long. So it's
No,
kind of a
no.
very scary thing to
Yeah,
jump
yeah.
into and get into.
Yes,
So
yes. In fact, when my lab went into it, I think they went with this dream, but they didn't realize what was the cost of it. And of course, we managed at the end that they're still doing it. It's just that, of course, that we lost probably a year and a half trying to optimize. And as I said, we come out from funding, and if you have a very you want to answer a specific question like this, as I mentioned, this guy that they want to do postnatal organoids, think about it first, because it might really, really, really hard for many reasons, not just for the organoids itself. For many reasons, it might be quite hard. Yeah.
yeah. Well, thank you again, Michela, for sharing all your insights and your expert tips with us. And thank you, everyone who joined the webinar. As I mentioned, if you do have any questions after this webinar is over, feel free to email us or Michela directly. And if you liked today's event and want to follow us on social media about any future events, we do post about them on there. So definitely follow us on our socials. So with that, I will go ahead and end the webinar, and thank you again, everyone, for joining. And thanks again, Michela.
Thank you, Fida. Bye.
Bye.
Bye.